Hello, welcome to the Cyrus Biotechnology demo for design tools. Today, I'm going to introduce you to how Cyrus Bench can be used to improve the stability of a protein system. To demonstrate the power of Rosetta and Bench, I'm going to apply several features within the Bench platform to a cytosine deaminase enzyme. I've picked this example because it is well characterized and was the focus of work that appeared in Science magazine. I've also picked it because the work was used to redesign a protein that is now in clinical trials. This is a very powerful demonstration of the kind of design work that's at your fingertips when using Bench. Here you see summarized the design work that will serve as the focus of this tutorial. In 2005, scientists at the University of Washington, including Dave Baker, and at the Fred Hutchinson Cancer Research Center in Seattle, published a paper in Science describing their efforts to thermal stabilize cytosine deaminase, which has been incorporated in retroviral gene therapy. The authors were successful in identifying 3 mutations that imparted a 10-degree improvement in TM while not affecting solubility or enzymatic efficiency. That same thermal stabilized protein is now in clinical trials for treatment of brain cancer, a major proof of the power of Rosetta and computational design. Here is the paper that contains the computational work that we're going to reproduce using Bench tools. Note that in 2005, this work was worthy of a science paper. Today, it's the kind of computational work that, with a little training, you can carry out in less than a day using Cyrus Bench. Of course, there are still experimental synthesis and assay aspects that will take much longer, but this demonstrates how substantially tools like Bench are changing the computational landscape. For the work I'll be showing you, I'll be starting with the published PDB structure with the ID IP60. This structure exists as a homodimer, and I'll be using that homodimer structure in the work I demonstrate in Bench. We're going to look for residues that can be mutated to improve the stability of the protein. So we're going to look at the positions that don't affect the ability of the protein to properly function or form the dimer, and also don't impinge on the catalytic site. We also are going to stay away from mutations that are mostly solvent exposed, and we do this for 2 reasons. First, they can affect the solubility of the protein in ways that are hard to predict. And also, predicting stability effects of solvent-exposed residues is fundamentally more difficult than predicting the effects on stability of buried residues. So to summarize that, we're going to allow mutations at residues that are not at the dimer interface, not within 4 angstroms of the catalytic site, and not on the surface. The color coding, as you can see in this picture, is such that the red residues are the ones that ultimately we're going to allow to change. And we'll use the design tools to figure out which of those mutations might actually be acceptable and ideal for improving stability. Okay, so this slide is going to show the workflow that we're going to use. We'll get the PDB file from the PDB repository. We're going to clean that structure up using the prepare process in Bench. Then we'll define the positions where we're willing to consider mutations, as I described in the previous slide. Next, we'll run the flex design protocol within Bench. This is a sampling method that tries hundreds of thousands of changes to both sequence and local structure at the mutation site, looking for changes in the sequence that may result in a more stable structure. One of the great powers of this approach is that because so many possibilities are tried, we can identify possibilities where more than one change in the sequence occurs simultaneously. Then we'll look at all the mutations that the sampling process has uncovered, both what residues have changed and what types of residues changes have occurred at those positions. We can use a logo plot for this. Finally, we'll use a method called delta-delta-G in Bench to triage all the possible mutations we observed to try to focus on mutations that are likely to be stabilizing. We're looking at changes that, on average, seem to be stabilizing in both chains of the homodimer. All right, so in this slide, this is just a little more background on Rosetta and the Rosetta scoring function. The reason that Rosetta is so powerful and so much better than any other software platform for protein design is the specialized scoring function that it uses. The Rosetta scoring function is a highly optimized combination of terms that are derived from the fundamental physics, plus terms that are derived from statistical analysis of known PDB structures and allowed rotamer analysis. This hybrid scoring function is both accurate and extremely fast, allowing hundreds of thousands of possible structures and sequences to be evaluated in a practical amount of time. This is in stark contrast to the energy function in most other programs, which is orders of magnitude slower to evaluate and therefore not capable of being used to evaluate enough combinations to provide reliable results for protein design. This is just showing the physics-based terms in the scoring function and then a variety of figures that show how the other terms in the scoring function are derived from the analysis of known crystal structures. Once we have imported the protein into Cyrus Bench, then we need to run the design protocol. And this slide briefly describes how design works in Bench. You define a sequence of interest, and that can be shown in this figure right here. And then the program will run hundreds of thousands of steps of random sampling to try to identify possible favorable residue mutations, while at the same time allowing the structure to move in the region of the mutations, both in the side chains and the backbone. When the calculation is done, you can examine the results both as individual best scored sequences, each with multiple sequence mutations, or as a logo plot, which shows how the variation occurs at each residue. So this is how the sort of bingo card, as I call it, is defined, and this helps us set up what kinds of residue mutations are suitable. This is the logo plot that come out of that in terms of what kind of variation did we see and where, and we can also compare the residues in a sequence viewer. Once we've carried out this calculation and we've identified potential mutations, we would like to validate those. And there are a variety of ways you might go about this. The way we're going to use today is shown on the next slide, and we're going to use something called delta delta G, which is another functionality within the Bench platform. So in the delta delta G, you basically bring in the information that you infer from, say, the logo plot. So what kind of mutations can occur at each position, and then you can run a free energy calculation, a delta delta G calculation, to determine the change in stability for each of those mutations. And what you're looking for is change in the stability that are negative, round zero or negative. And that suggests mutations that will be satisfactory and can be accommodated by the protein without destabilizing it. If the delta delta G is large and positive, those are typically not mutations that are suitable for design. Now, I should note that because design itself allows multiple simultaneous mutations and the delta delta G procedure does not, there are cases where you can get non-additive effects from multiple mutations, and you won't pick that up with the delta delta G validation. But delta delta G is pretty good at identifying mutations that are very large and positive, and those are ones you may wish to triage away. All right, so that ends the introduction, and so now we're going to go to Bench itself. All right, welcome to the CAD portion of Cyrus Bench. I'm going to assume that you're already familiar generally with how to log into Cyrus Bench and that you've brought yourself to the CAD portion. So the first thing we need to do before we can do any design is we have to import a structure that we can work on. To do this, we will have clicked on the structure loader button here, which brings up a panel exactly like the one you see here. And then we're going to upload a PDBs file. And as I mentioned, I'd already created a PDB file that we can use with this project. I've modified the crystal structure slightly by removing a few residues at the beginning of each of the two chains that are, according to the paper that we're following, simply clonal artifacts. This results in two chains, both of which start with the sequence MASK, so the sequence and residues. So we're going to bring that in. You'll see I double clicked on that, and we'll only have to wait a few seconds for the program to import it. And in the import phase, it's assigning parameters and calculating a score. See how fast that was? We're already done. If we go to the left and click on this new line here, which is the import, we can see that the structure has been brought in and it has been assigned a Rosetta score. Now this is amazingly fast. If you've ever used another computational platform, you'll know that most programs take quite a while to calculate a score for a protein and another good amount of time to assign parameters for it. And this is just by comparison an order of magnitude faster. Now there are a number of things we can do when we bring in a structure. We can look at it by clicking this little eyeball right here. If we click that and we go down to the bottom and we make sure we auto zoom, We can see we have the structure right there, and that's the homodimer. We can look at the sequence for the structure if we wish by clicking on this upper left-hand corner button here, and that brings up the sequence. We can change the relative sizes of the different panels in this view by moving this bar here. So since we have no other information up here, we can have the picture of the molecule take up more of the screen space. Another thing we can do is do color coding. So for example, if we would like to see the two chains in different colors, we first change this drop down here to chains, which means now our selector is at the chain level. Let's pick one of the two chains here, and we can choose this button here to change the color. Let's change it to yellow. All right, so now it's very easy to see the two chains in the homodimer. It's important to note that the numbering convention in Cyrus bench follows that of Rosetta. What that means is that the first residue in your system is always assigned residue number 1 as far as bench is concerned, and that all the chains are concatenated. So if you have two chains, as in this case, the residues of chain A are shown from residue 1 all the way up to residue 153, and immediately after that, residue 154 is the first residue of the second chain. This will become important if you're trying to follow the literature where the protein numbering follows the standard numbering rather than this concatenated numbering. So you will have to figure out that correspondence. I've already done that for the demo I'm going to show you. All right, now that we've brought in a structure, the next thing we have to do is prepare it. Prepare does an aggressive minimization that's constrained to the starting structure. It's required before you do any additional design work. It can take anywhere from a few minutes to an hour, depending on the size of your system, but you only have to do it once. To run it, we simply select the input structure by clicking this box, and then go over here and click on the Prepare button. And then we click the Run button here, and the program will go off and prepare this structure. And this can take anywhere from a couple of minutes to maybe 20 minutes. And through the miracle of pausing, we'll come back when this is done. All right, and what you can see now is that two things have happened to this screen. First off, you can see that the job that we just ran shows up here with a check mark, meaning it's done. And over here, the results of the job are shown, and the little new button is showing that these are brand new results. So we now have a prepared structure. To look at the prepared structure, we just click on this line over here on the left, and this brings up the prepared structure. You can see that we've assigned a score to it. One more thing I haven't noticed is that we have all the contributions to the score over here on the right-hand side. But generally speaking, for especially when you're starting out, the main thing you're going to focus on is the score right here, the total Rosetta score. If you want to see how the score has changed relative to the input structure that we started with, there's an easy way to do that. We can click this Compare button right here, say that we want to look at the imported structure right there. And now you can see here's the imported structure, the PDB structure, which had a Rosetta score of -293. And here is the prepared structure, which has been cleaned up and has a Rosetta score of -392. That's typically what you would expect. You would see that the score would be reduced. All right, so now comes the fun part, design. Design in Bench is an approach to identifying changes in sequence to improve affinity across an interface or the stability of a protein core. In our case, we're going to focus on the latter. It does this by a random walk of changes that simultaneously vary sequence and conformation. There are two kinds of design currently within Bench. One is called just design, and the other is called flex design. The difference between the two is that flex design allows more movement of the backbone during design. Both methods allow sidechain rearrangement, and neither method allows drastic backbone motion. The most movement that you'll see with flex design is maybe a few tenths of an angstrom, but that's enough for many design protocols, and certainly for the type of design that we're doing today, which is to improve the stability without affecting the ability of the enzyme to do what it needs to do. In order to do any advanced modeling within Bench, there's one thing we have to take care of before we can actually do the calculation, and that is to create selectors. Now, a selector is a group of residues that you're going to perform an action on. And the nice thing is that we can create these selectors and reuse them as many times as we want. We just have to create the selector once. To create a selector, what we would have to do is we bring the structure into the workspace, as we're going to do here. Now, we then can select residues in the structure and create a persistent selector. Just to give you an example, let's say we were picking at the chain level and we pick this chain here. Now we've selected half the protein complex. And to create a selector for this, we go over here and click this Create button. And now we've created this selector 4. And if we push this button right here, we can edit it. And you can see here's all the residues in chain A of the protein complex, and they're all selected. If you wanted to change this, all these positions can be turned on or off. So if I click here, it turns it off. If I click again, it turns it on, and so on. So this is how you would generate a selector, and you could have as many or as few residues as you want in a selector. Now let's go back to the prepared results again, and I'll show you how to set up a flex design experiment. All right, once again, these are our prepared results. As I mentioned, I had already predefined some selectors that can be used for the calculations we wish to do. These selectors are based on the selection of residues as I described in the first part of this presentation. In other words, we're selecting residues for possible mutation where the residue is not involved in the binding site, it's not involved in the protein-protein interface, and it's not on the surface. So this selector is given right here, chains A and B, no surface, no interact. And if you rotate this around, you can see that those residues are in fact mostly or all in the core of the protein complex. So we're almost there. Now we can run Flex Design. All we have to do is select this prepared structure, click the button here, and click the Flex Design button. And this brings up a panel where we have to provide a little bit of information, but not very much. The first is we have to provide the selector. You'll recall that the selector, which we predefined, determines which residues will be allowed to mutate during the design experiment. So we already have these predefined selectors, which we've discussed, and we'll pick once again chains A and B, no surface, no interaction. And you'll see that we get this list, and it's a very, very long list. In terms of design, that's a lot of residues. Next, we need to decide what kinds of mutations are we going to allow at each of those positions. Generally speaking, when you're doing design work, you're usually happy to, in the first phase of your experiment, to let all types of mutations occur at each position. To set that up, you would just click this upper left corner where it says POS, and you can see that all the positions light up. That means when we're doing the design experiment, all mutations at all sites are on the board as possibilities. Now, in the paper we're trying to reproduce, they actually did not let cysteine mutate, so we're going to unselect that. If in more general case you were doing a design experiment and you were interested in not allowing, say, certain types of residues at specific positions to not be mutated, you could pick those in this bingo table as well. For example, if I didn't want to let isoleucine occur at position 16, I would click that and it would turn it off, and we can turn it back on by clicking it again. So you have full control over this bingo table. In the case that we're going to deal with today, we'll let all the possible mutations except cysteine occur. Okay, so in this panel, the only other thing we need to specify is the number of repeats, and that can be specified in this box right here. Each repeat represents an individual full calculation, only with a different random number seed. By repeating the experiment multiple times, we get a good indication of which residue positions have a tendency to mutate and what residue types are tolerated at each position. Because of combinatorial explosion, typically, no one calculation will give us the singular best sequence. Even nature, with affinity maturation, is not efficient enough to do that with such a broad number of mutation sites. But by running many times, we get a very good idea of the extent and variety of possible mutations. It's worth noting that with other software packages, you'd have little hope of being able to explore enough sequences or conformational space to even identify something close to the best mutation set. But because Rosetta is so efficient, we can here. And the other thing worth noting is that because Bench uses the Google Cloud, on the back end, you have instant access to a very large array of processors, allowing you to run these calculations in a hyper-parallel fashion and get back your results rather quickly. So we'll set this to 100 here, and then we'll click the Save and Run button. and the calculation will go off, and through the beauty of time lapse, we'll come back when the calculation is complete. Okay, and we're back. That took about a half an hour to run. And as you can see now, we have the Flex Design results over in the top left-hand corner. So we can now click on that collection. And what we see now is the set of results. So there are actually 100 structures in this list. And we can zoom down, scroll down and see all those 100 structures. And this corresponds to the 100 separate calculations that we requested. We can sort by score. So now the best Rosetta score sequences are at the top of this. And if we want to look at the different structures or possibly look at their sequences, we can click on some of the eyeballs. So if we click on the eyeball there, that will show up if we click on let's just click on the eyeballs for the first 5, the best ranked sequences. And you can, just looking at the structures doesn't give you a huge amount of information, but we have the ability to also look at sequence information. And to do that, let's compare those to the sequence of the starting structure. So now we have the starting structure is here. These are 5 of the best sequences that were determined during the design experiment. If we click on this button right here, we'll see the sequences of all these structures. And if we click on this button right here, we can see where sequences vary with one another. So this is a way to kind of quickly identify where these mutations are occurring in the different sequences. A perhaps more striking and immediate way to look at sequence variation is to look at a logo plot, and we can get that by clicking on this selector down here in the bottom right, and we pick sequence logo, and here's the logo plot now for our experiment. And we can click this button right here to the upper left of the logo plot, and we can focus our interest only on the positions where we allowed mutations to occur. So if we scroll through this, we can see that in some positions, really, there was no variation. In some positions, there was a modest amount of variation. We're not seeing a huge amount of variation anywhere, and that's probably good. So what this is basically letting us see is that we do have some variation at a small number of positions or a modest number of positions, and this is going to make it much easier for us in the second part of our design, where we have to really focus on what types of mutations that we would, if this were a true experiment, we would send to get synthesized and assayed. So seeing the information in a logo plot is good. Actually looking at it in a sequence viewer and highlighting where all the mutations occur and what types of mutations are seen might be a little more straightforward. And to do this, to see this in a sequence viewer, you actually have to use an external sequence viewer. Use the one that whatever one is your favorite. To export all the sequences that were determined during Flex Design, you would first select all the residues by clicking this box here, and then we can download. These are now all selected in this table, and we can now click the download selected button here and export those as FASTA sequences by clicking here. So I've already done this offline so I could show you the results, and here you can see this is the analysis looking at a sequence viewer for the native sequence is shown in the first line, and then some fraction of the design sequences underneath that, and I'm color coding wherever the design sequences are different than the native sequence. And you can see that for most of the positions that we allow design to occur, there really isn't any variation. The native sequence seems to be the best, and for a very well-evolved protein, that's actually not unusual. And then we see for certain positions there are in fact mutations that are seen over and over again in Flex Design. So this information that we see in this table can be used to do some additional analysis. And to start this out, you would create a selector that focuses only on these positions where we do see some modification in the first round of flexible design. So let's go back to the program. And what I've done, and I did this again offline in the interest of speed, is I've created a new selector which I call Chains A and B Round 2. And this really just focuses the effort on the mutation sites where we did see some variation during the Flex Design experiment. Now, what can we do with what we have so far? One thing we could do is look at the sequences that were designed and see what kind of mutations are in each of those. And in a real life example, you would combine that with your knowledge of the protein system and your knowledge of protein design and say, which of these seem reasonable and which of these maybe don't seem as reasonable. But there are more automated ways of looking at this data. One of the ones, and this is a really nice way of looking at the data, is to look at all the mutations that we saw in Flex Design and see which of those, if we do a free energy experiment, a DDG, which of those are individually seen to be stabilizing to the complex. And so I think that's what we're going to do next. And in order to do that, once again, we're going to start with our prepared structure. So we'll click over here, and it brings up the prepared structure once again. And now we're going to click this DDG button here. And DDG, as I've described earlier, is a way to calculate the free energy for any single mutation in the protein. So we can ask for a variety of different types of mutations, and it will individually make each of those mutations and calculate the free energy for that mutation, which gives us a sense of whether it's stabilizing or not stabilizing to the protein. So let's use the selector that I created for round 2, which is chains A and B round 2. And just to clarify again, these are the only positions where we saw changes in the sequence in the Flex Design experiment. Now, what we want to focus on are the changes, the particular residue changes we saw at each of those positions. And I've made a selector, a mutational selector, that's based on that analysis of that table I just showed you a second ago. That would be this table. And I'm going to upload that now in the interest of speed. So that would be here. All right, so this table encapsulates all of the mutational information that we saw in that analysis of where the mutations were and what types of mutations occurred at those positions. So what we can do with this now is run a free energy calculation to determine what the free energies are for each of those individual mutations. And from that, we'll be able to prune down this total list of possible mutations to a more manageable number. Before I go with that, it's probably worth noting that even if we had to focus on all these possible mutations, compared to the number we started with in our initial set of possible locations and mutations, this is a tremendous reduction in the complexity of the problem. But let's run the DDG calculation and make our life even easier. We're ready to run this, and all we have to do is click this Save and Run button, and this will take a few minutes to run. We click the Start button, and all those green boxes turned into gears, which means that now the calculation is going to start. And again, we'll pause, and when the calculation is returned, we'll return to the presentation. Okay, and we're back. That took less than 10 minutes. So that just, again, shows you the power of parallelism of the cloud computing plus the speed of Rosetta itself in performing these types of calculations. So what you see here then are the free energies for all these types of changes. For example, at position 9, changing glycine to an alanine, it gives a positive free energy in terms of stability of 4 kilocalories per mole. In other words, pretty destabilizing and something you'd want to avoid. And similarly throughout this table. Once again, I'll emphasize that this is for the homodimer protein. One thing that I haven't mentioned yet too much is the fact that in the Flex Design experiment, there is no accounting for the fact that this is a homodimer. In other words, every residue position is independent. And because of that, and because it's a stochastic process, you will find situations where a mutation is, say, seen in chain A, but not seen in chain B. You won't see necessarily the full symmetry occurring in the results that you find. So the free energy calculations that we're doing here now are not only a good way to sort of validate the mutations that Flex Design is suggesting, but also a way to look and see if those mutations are really suitable in both chains of the protein. And I would also add that since this is a crystallographic protein structure, there are modest differences in the two chains conformationally. All right, so I've got this table now of the mutation data, the stability calculations for all the positions where we saw changes during Flex Design. And for the rest of this analysis, I'm going to go to a set of slides that work off this table and are just, it's easier to present it in that form. Okay, so this is the matrix that you just saw in Cyrus bench, now brought over to a slide. And the slide is just to make the discussion that follows a little easier to follow. So you'll see a variety of values, and they range from quite large, say 7.5, to quite small, which is maybe 2.5. These values should qualitatively tell you which positions are going to be favorable for stability, the ones that are negative or not much above zero, and which ones are positions, which are mutations you probably want to avoid. Now, I'm going to make this a little easier by doing a little bit of annotation. All right, so I've drawn a heavy horizontal line here to separate the A chain from the B chain. As I mentioned, if everything were ideal, these values for the A chain and the B chain would be identical, but the crystal structure itself is not fully symmetric, so you see some of these values are larger in one or smaller than the other. So now I've done an analysis where I went through and put a box around every position, every mutation, where either the A chain or the B chain had delta G that was less than 1. So if the A chain had less than 1, but the B chain was greater than 1, or vice versa, that was sufficient to get a box around it. So these are all the positions where either the A chain or the B chain was less than 1. All the remaining mutation positions that you see there with no box around them are probably positions that you don't need to consider very much, at least in your initial rounds of experimental design. The next thing I did was average the values for the A chain and the B chain for the positions where there's a box around them. And in situations where the average was less than 0.5, I changed the color to green. So these are positions where the average for the mutation calculated in the A chain and for the mutation calculated in the B chain were less than 0.5 and are probably reasonable to consider for design experiments. So you can see that even before I made that distinction between the most favorable and the not as favorable positions, we had already reduced this to a pretty manageable design experiment. The amount of information contained in 13 positions for possible mutation is enough that you could really go forward and probably make a good portion of the possibilities there, at least one-offs experimentally. But let's really focus this even better by focusing now on these positions that are marked in green, which are the most favorable on an average basis mutations suggested by the results of flex design. So one thing that's very interesting about this is that if we continue on the analysis here, now I've just removed the bottom part because it's not really necessary. It's symmetric in sequence, and I've done the analysis in the top part. Now, this is analysis that obviously you couldn't do without having experimental data, but looking at the experimental data in the paper, it was clear that 3 of the mutations that previously were shown here in green, this one, and this one, were actually mutations you would not want to carry out because they had very deleterious effects on the solubility of the protein. So again, it's cheating a little bit because we had to look at the paper to find that information out, but it's clearly information that you would determine in a real design process. So that then leaves 5 green boxes, 5 positions where you would want to look at the residue mutations based on this flex design and DDG analysis. And obviously with 5 possible mutations, you really could run through the combinatorials of that and look at all those positions 1 at a time, 2 at a time, and 3 at a time. Now, what you will find if you were to do that, and this is the most interesting thing of all, is that 3 of those 5 positions where the flex design combined with DDG says you want to focus your efforts, those represent the triad of mutations that in fact in the paper they found were suitable for creating a protein with an increased TM by 10 degrees, a 30-fold increase in half-life at 50 degrees, and no reduction of catalytic efficiency and solubility unchanged from wild type. So let's just summarize this now. I ran flex design. I ran a delta G experiment to triage through the mutation suggestions from flex design, and after doing that analysis, I found that 3 of the 5 remaining positions, in fact, corresponded to the 3 positions that were used to stabilize this protein and were later used to create a clinical candidate structure, which is now in phase 2 clinical studies. So I think this is a pretty impactful and impressive use of the bench protocol to design a stabilized protein. So at this point, there's just one last thing I'd like to show you, and that is a structure with the 3 mutations in the structure. So I've created a PDB file where I've made the mutations that are shown here, the 3 mutations that were discovered in the paper and then are in the clinical candidate, and that we discovered here, which is the alanine to leucine 23, valine to isoleucine 108, and isoleucine to leucine 140. One more point I'll make here, because I think it probably needs to be made so no one gets confused. This is residue 23 in the PDB numbering. It's residue 18 in the bench numbering. This is residue 108 in the PDB numbering, and 13 in the bench numbering, and this is 140 in PDB numbering and 135 in bench numbering. This is because the numbering in bench starts from residue 1, whatever residue that happens to be, and not the PDB numbering in the paper. So just in case there's any confusion. So let's go now back to the live bench demo, and I'm going to show you the structure that I've already imported, which has the 3 mutations in it. And that structure is shown here. Again, it's the homodimer. It's very obvious what's going on here. And then the 3 mutations in each of the monomers can be seen as we scroll around a little bit. So you have 2 residues that moderately interact over here, and then the 3rd residue is down here. And that's repeated on both sides of the dimer. So that about does it now for the demo, and I'd just like to thank you for your time, and I hope you found this useful, and we'll see you for the next demo.